headphones. Sorry.
Oh yeah, Andy, were you able to highlight that video?
Okay.
You can still.
Good, I did not.
Oh, thanks.
All right, I'm going to try and wiggle that thing a bit.
Sorry? I don't know why it's... Okay.
You
can
keep
make it
on
work.
them.
Yeah.
Vidita, you keep on them. You guys slacking over here.
Well, it's
How do you have some
very
momentum?
exciting. The excitement sometimes
Uh-huh.
makes you
We're
forget.
going to call that the bump and slurp.
Yeah, I think the viewers have been counting the number of bumps that Trevor has been doing, and
We got a
he
bonk
loses
tally?
points, and
Oh, yeah,
and
bonk
now
tally.
he got them all back with this.
I try to announce all my bonks. Just try to be, you know, open and transparent about these things.
Can I grab bubble for a minute?
Grab it.
Yeah.
Tunicate, another stock tunicate. Something down there at the base. I guess it's another tunicate.
Something down where?
At the
Where
base of
is
the
that?
rock. It's just this thing. No, it's a set pocket. Never mind.
Oh,
I do see a little stick thing, though, a sticky stick thing.
That's something.
Zoom in on the sticky stick.
Yeah,
Couple,
okay.
couple 3 sticks, actually.
Oh, jeez. Oh, those things.
What are those things?
Sticks.
Oh, okay, roger. Come wide on the sticks.
That's some nice sediment there.
I don't think those are cladorhizid. It's about the size of some cladorhizids, but usually they have a little more definition to them.
What's this red thing?
Black coral.
Yeah, black
Yeah.
coral.
Black coral.
Yeah, the old Staropathes is what we're going to on these guys now.
Oh, I'm going bathy-pathy.
No.
Okay,
Zoom in, please.
challenge is on. Oh, and Trevor strikes out.
What?
Yeah.
I actually don't know how to tell the difference. I thought there was more arms on the Staropathy.
There are, but the translucence, you see it's almost like it's
Oh,
luminescent or something.
Almost like anemone arms.
Yeah, that's a clue, I think.
That's a Staropathy?
I think so.
Cool, thanks. Learning.
Even if we're not seeing those secondary branches.
Oh, just because it's young or small or whatever.
Yeah, younger, smaller. I think they have to get to a certain size, but
The bonk and slurp. Yeah, it was effective. But it couldn't happen without Antonella with the assist there. Had to remove some arms so it fits.
That crinoid didn't know what hit him.
Oh.
Ooh, little, what is that? Nothing. There's another one.
this is definitely a more interesting dive than I thought it was going to be.
Yeah, it's gotten better.
Yeah, very productive as well.
Mm-hmm.
A lot done.
Sampling, yeah.
Well, we're already well past our end point, so we're covering a lot of
Can you zoom in,
ground. We're
please?
getting a lot of good close-ups,
Zoom, zoom.
getting animals,
Tunicate.
tunicate.
Ooh, nice eyes.
Okay,
Well,
thanks.
it's better than 3 sediment patches, which is what I've called out. Jeez. That could be my record for identifying sediment patches.
He had the sediment philia bad this
Yeah,
morning.
really bad. Is there an antibiotic for that, or
There might be. I think bacon.
bacon?
That's my, I think if you eat sufficient amounts of bacon, you will alleviate the,
sediment philia?
uh-huh.
Do you think there's going to be any tater tots this morning?
You kidding me?
I hope so.
The ship would collapse without tater tots.
I love tater tot day.
Every day is tater tot
I
day.
love every day.
My favorite is walking past the table after they're put out and just grab one and grab and go.
Oh yeah, the table tater tots.
I noticed the reappearance of the table bacon the other day.
Oh yeah, table bacon's a classic.
It was just like a
one-day appearance.
Weird.
Can our viewers tell it's breakfast time down and after? It's 2 minutes past the breakfast time for.
Where's our breakfast relief?
I know. I think they're gonna let us run out the clock before they feed us.
They think we've been having too much fun.
Or
Absolutely.
or just snoozing.
Yeah. By the way, front row, Sako Matsumoto actually made that comment. Great fun watching all of this this
Oh,
morning.
awesome.
Bridge nav, another 20 meter 075.
Do you have like a highlights collection skills highlights video reel?
I mean, Trevor, right? You're asking.
Yeah, Trevor.
Do I have one? No.
Or I mean,
Like a
does
personal highlights
Nautilus have
video.
one?
Oh, you mean like, do I ask Justin to pull me a snip of this video at the end of it and say, look at this sample?
You
I
should
think
definitely do that with some crazy music.
yeah.
You could just like have it.
Some, like
So
heavy industrial music, you know, or
I have made cool animal highlight reels on other ships with other organizations that we have more easy access to the video. That we're actually like ROV departments, the video providers as well. So I was on a Coast Guard ship earlier this year, Canadian Coast Guard, and the Coast Guard deck crew was the one running the winch, and they run all the deck ops, and they were fascinated with all these animals they hadn't seen before. So at the end of the cruise, I put together a little 3-minute kind of highlight reel for them, and they got to see, you know, fish and sponges and all the sorts of cool stuff, and they loved that.
That's pretty nice for you to do that. That's great.
yeah, that's great. Well, I think we need a highlights the kind of a Olympics of collections skill video.
I applaud any and every effort to generate interest in the deep sea by anybody. It's just, you know, I mean, by anybody, targeting anybody, whether it's other engineers on a ship or kids at school or adults at home or businessmen or anything else. Any way that we can get people to be interested in this environment is a really good thing, if you ask me.
I taught one of the ABs on that Coast Guard ship the term compound tunicate and didn't tell anyone else on deck. So they have a little video feed out on deck so they don't have to stand around for 8 hours just bored. They can actually watch the video feed. And one of the bosun says, "Oh, look at that thing." And AB says, that's a compound sea squirt. And he's like, "What?" No, it is. So proud of himself.
We were, for Thunder Bay National Marine Sanctuary in May this year, we were also running OET expedition from a Coast Guard vessel, and they were also like, said it was the coolest thing they've ever seen. They're just usually tending buoys and stuff, and we were diving on shipwrecks all day. It was great.
That's cool.
Bridge and nav, another 20 meter 075.
It's cool to give those people that option. You know, people are out on Coast Guard ships because they love the ocean in some capacity, and they, you know, see all sorts of cool stuff, a bunch of different clients or whatever tasks they're doing, but not often is it ROV. So they get to see this thing that they spend a lot of their life on from a whole new perspective.
Yeah.
It gives me a new insight on what they're actually doing it all for.
Yeah, we have seen all kinds of rock formations. These rocks tell a story. It's very interesting to just to see them at the bottom of the ocean and then come up to the ship, and you can actually get to touch them and even smell them.
Mm-hmm.
And we don't even know what kinds of microbes are carried in these rocks.
There is so much to learn down here. It's crazy. And Beth is kind of a pioneer looking at the microorganisms that are down on these rocks, because that's so difficult to do if you think about it. So she really is working on opening up a whole new field, her and her colleagues.
And you never know what you're going to find, right?
Yeah.
Rocks, geology, all kinds of biological communities, species that have not been discovered before, and microbiology, and even man-made things.
Do we have room for one more rock?
Only always.
Because, I mean, I just worried they're going to have a time next watch.
Yeah, we can't let them have time, you're right.
Yeah, so
Go ahead, get the arm out.
We've done so well on this dive and got all of our objectives. We can spend
rock
some
sample.
time in helping Andrea out and getting
Keep eyes on it before you move it.
There's a nice angular one, just lower to low.
Where is it?
Below center, to below and right to the center.
Are you looking at that one?
Below and right center, I see it.
That one, yes. We had the rock consensus there.
Is that too big, this one, though?
What am I hitting?
Rocks.
I think I'm hitting rocks. Oh, there's a rock right now.
Where do all these rocks come
I'm
from?
stuck. Unstick. Oh, you want to do that one?
Yeah, it isn't too big, is it?
No such thing. Come on.
That one is nice rock. That's a good rock.
That is a good rock.
I'm going to winch down a little.
Yes, curious minds want to know how old these seamounts are. Current thinking is that they're 80 to 100 million years old, Cretaceous era origin.
And we talk about, you know, the rocks for this person, a rock for that person, but really it's for everybody.
It is. It's for the
gotta
world.
Be available to everybody. Come on. Don't do that.
Nah, it's gonna go.
Is it?
It just that the claw was oriented.
Yeah, it'll go.
Yeah.
I see it wiggling.
Yep.
You do? Okay.
I see it wiggling. It'll go.
So when this volcano was erupting, this seamount was quite a bit further south. It was in the equatorial, Cretaceous era equatorial.
Okay, get out of there a sec. I'm going to
likely.
reposition. Okay, go for it.
The Earth was very different back then.
Yeah, nice.
Yep.
Broken free now.
Yep.
Can't see.
Nice.
Nice blind grab. No big deal.
Oh, all right, Antonella.
Yay.
Hey.
Oh, you got a twofer.
Oh yeah, bonus rock. Nice.
Bonus rock.
Where?
That one doesn't count.
Right
bottom.
below.
Oh.
It's mama and baby rock.
That's really
Showing
funny.
it off.
Oh, that looks good.
Nice
Wow.
job.
Oh.
Look, 2 different life
Beautiful.
stages of a rock.
Yes. Oh, look at that nice,
Nice.
yellow
Very nice.
iron.
All right,
And
starboard
you
box.
Box of rocks.
Box,
Which
box.
Where's it?
We got
Let's go into Charlie. There's a challenge for you.
Charlie's
Put it
open.
in the small box. Why? Samp. Nothing floaty over here, is there? Nah. Made it hard for you by extending the box early.
So, Kainalo, have you smelled the rocks that come into the lab?
Is he
What
gonna,
do they
is,
smell
I-
like,
I
is he going to
Chris?
admit it? I don't think he's going to admit it if he has.
Well, it's so cool to
Why
use
not?
all of your 5 senses when you are exploring.
I thought
Yeah,
I saw him
some-
down there one night about 3:30 in the morning, very secretly sniffing something. Have you smelled him, Kainala?
Yeah,
We
they
got the small rock in box Bravo.
box Bravo. Okay.
The big rock is perched on top. It'll fit, it just needs a little rotate.
So when this seamount was formed, there was a huge gap between North and South America, or what is currently North and South America. There was also the huge inland sea that covered most of the central part of the United States. It's really fascinating how the Earth, the continents have changed over time. Oh, and their tentacles retracted on that.
Yeah, I bumped them.
Nice.
What is this storm all about? Starboard side storm. Maybe it won't go.
Oh no, it's gonna go.
Oh.
Needs a little nudge.
I think it needs a little
I
nudge.
don't know. That's how we
And
lost that Sea Star the other day. It was created a gap in the box.
Yeah, don't want a gap in the box.
We've
It needs a
repaired that
little
seal
bump.
too. We fixed the seal, so it should go.
Oh no.
Oh, you did? Okay.
Yeah. Nice.
There you go.
Nice.
Nice job, Antonella.
Woohoo!
Way to go.
Nailed it.
3 points.
Nice.
Nailing it on high pack over here.
So the big rock was in box C.
That's correct. Yeah.
Thank you.
Hello. Bye. All right, dive salvo.
That crinoid is really disappointed. It's going, man, I had this huge food particle. It was all yellow and everything. And I just almost had it.
The one that got away.
The one that got away.
It was this big. Oh, I should probably bring that box in, shouldn't I? That's what we do here.
Someday you're the predator and someday you're the prey.
That's lovely. Bonus rock. That's hilarious. I know it's totally blind. That was also really good. You just gotta go off memory. It's not easy, but.
Oh, we have a viewer here, Gina.
Aloha, Gina.
Hello, Gina.
She has been inspired to create crochet replicas of some of the amazing deep sea critters.
Crochet
She's seen-
replicas.
Oh,
Yes.
cool. That's great.
Zoom in on this thing,
And
please.
right now she is crocheting
Anemone.
a few googly-eyed squid.
Oh,
Nice.
awesome.
That is cool. Way to go, Gina.
Crocheted anemone. That would be
Just
kinda
finished
cool.
a few gulper eels,
Ooh.
piglet squid,
Nice.
Send us pictures.
and flapjack
come on, please.
octopus.
Very
And all the
cool.
highlights.
Thank you, Gina. That's terrific.
That's amazing.
You know, I was on a cruise a while back too, and we saw one of these finned octopus, which, by the way, have not shown a show. Have not had a showing here, and I'm disappointed. Where are all the finned octopuses? But that inspired a young lady who was on the cruise with us to get a finned octopus tattoo on the back of her neck.
Oh my goodness.
It was a very cool tattoo.
So apparently, she knows people who have been on the Nautilus because they have placed orders from her.
Really?
Yeah. Well, thank you, Gina, for
thank you.
sharing such a beautiful experience.
What form does the gulper eel crocheted have? Is it
Well,
like is it a
probably the
scarf? Is it a hat?
I don't know. I wonder if she has an Instagram where she posts all of her
It's
pictures.
probably a plush toy,
A plush
That's
toy?
my guess, yeah.
Oh, okay.
like a crochet.
What's the
toy.
matter? I missed it. Oh.
I think it'd be great as like a gulper eel shopping bag, you know? or it's like squished down to this tiny little thing, and then they're like, you can't fit all this stuff in there, and you're like, yeah, watch me. Watch me. I'll put everything on that conveyor belt in this bag. Be the ultimate bag of holding.
We have a question from friends.
Okay.
Have you seen invasive species resulting from accidental human importation?
Not this deep. Hawaii in general has suffered from that problem. There's been a lot of invasive species, and gives Andy a chance to talk about the measures that the monument management take to try to minimize invasive species in the monument.
Is that a softball for me?
That's a softball for you.
I'm excited. Yes, we are super serious about our invasive species in Papahanaumokuakea. And for the marine realm, what we do is we investigate the hulls of any ship that's going to come up here and do work. So we actually dive on them, or sometimes send down a little ROV to look at the hull of the ship and see if there's any barnacles or hydrozoans or other things that are attached to the ship. Basically, if they are, we ask them to clean them or have it cleaned before the ship comes up here so it doesn't introduce any new critters into this environment. And for some of the shallow water work, we even cleanse our scuba gear and all our gear between islands so that we don't transfer species from one island to the next. So that's after they come back from a dive, like on a coral reef, we'll put the scuba gear, scuba equipment, and fins and mask and everything in a light bleach solution that will kill anything and not allow you to transfer between islands. Because right now, actually, we are experiencing a severe outbreak of a algae at Pearl and Hermes that has taken over quite a large part of the reef. And it's actually a new species to science, and we don't know a lot about it, and we don't know how it got there. And it was also found on Midway more recently. So it is a big concern, because once an invasive species gets in the marine environment, it is next to impossible to remove it. And on land, we've done the same thing. If you go onto any of these islands to do work, you have to have all new clothing frozen for 48 hours, put in special buckets, and only put on when you go onto island. And each island has its own set of clothing you can't use between islands. And that's to try to reduce any introduction of insects or seeds or anything like that.
Does the freezing actually kill the seeds?
Yeah, the freezing can, well, this is part of the reason why we asked for the clothing is completely new. But yeah, the freezing can make them so they don't germinate, but not always, as far as I understand it. The more the concern is the insects.
And that's another one, is do the insects die from freezing?
Yeah, the insects, it will kill the insects. That's why it's 48 hours.
So what if it's an insect that handles, like a lot of insects do handle freezing, mosquitoes, or one like, the Arctic is full of insects, but how do you know what insect will survive?
Well, a mosquito won't survive freezing out in the open. Its larvae may be in the soil, which has its own kind of heating, from
Does it?
decomposition. Yeah.
Even in permafrost? Like I'm talking Arctic, right?
Uh-huh, yeah.
So where's the heat come from?
Oh, just decomposition. I mean, it's not quite like...
Of something they're eating or something.
Yeah.
Are you guys noticing these very interesting patterns in the sediment?
Mm-hmm.
Oh, you can count on Andy noticing that while he's talking, because he's a sedimentaphiliac.
I am a sedimentaphiliac.
Do we need some pictures there for Andrea to analyze? For our geologist?
Core? Are you talking about a core? I am.
No, she's talking about the imagery for that manuscript that Andrea's talking about those internal wave.
Oh, I don't think so. I don't think we see any. I don't see any of that here. Do you, Chris?
Yeah, there's some ripples back there, but I don't understand how you tell how what type of wave created one type of ripple or anything.
I got a good description of that yesterday, so I'm going to try to describe it. So in the sediment, we have seen some wave patterns, looks like from either current moving or what we call these internal waves, and it's basically ripples in the sediment. And the difference between those that are created by currents is that a current one is very uniform and the wave is not even on both sides. It actually looks like it has a little bit of an arc to it at the top, and they are uniform across the entire wave pattern on the bottom and across the entire wave and crest pattern on the bottom of the sand, whereas these internal waves, which,
brittle star.
We have biology.
That's really far away.
Yeah, brittle star.
Whereas these internal waves, which are produced from the dense water moving up and down from reflections of energy off of these seamounts, actually produce these very uniform
Look at how cute
waves. So the trough and the crest of the wave is uniform in this kind of sine wave pattern.
Yeah. Look at this cute crinoid.
Very
this
cute.
could be a different species.
Okay, zoom in on the cutie.
Cute crinoid. Disrupted my wave talk. Very distraught.
Oh, it is very bonky down here right now.
Man, it's cool. Everywhere we look, we see those little sea stars, those brittle stars, Chris, the little one on the right there.
Yeah. So checking the crinoid out, this is probably Hyacrinidae, so it's very different than the one we collected, for example. But yeah, that's great.
Okay,
Thank you
thanks.
very much. It's a lighter colored yellow, but all the Hyacrinids that I'm aware of are yellow.
That was a 5-legged crinoid too.
Yeah, and that's another characteristic of that family, five arms rather than legs.
A viewer is asking what is causing the surging that Argus and Hercules are experiencing right now.
Well, Hercules is me flying poorly and hitting stuff.
Is the surface wave acting being transmitted down the umbilical?
Yes, so Argus is connected to the ship with a big cable that's under a lot of tension. Argus is actually about 3,000 pounds?
Yeah, 3,500, yeah.
3,500 pounds negatively buoyant. So it's basically a big weight. And as the ship heaves up and down, Argus is moving up and down with the ship. So when we're experiencing, you can see it in the Argus cam, the sort of how calm the sea is a little bit. But because there's a soft tether between Argus and Herc, we try not to transfer all of that movement to Herc, so Herc can be really stable for manipulations and that kind of stuff.
Well, to our viewers out there, thank you for following us on nautiluslive.org. We had a very exciting watch, 4 to 8. Thank you for your comments, your questions. Keep them coming. We are going to transfer to the next watch.
Thank you, everybody.
Yeah, mahalo, ahui ho.
Thank you.
Check, check, check.
Morning, everybody.
Good morning.
Good morning.
Good morning.
Let's do this. All right. Well, as we do on the beginning of our watches, we're going to take a moment to kind of recenter and ground ourselves in the work that we're doing. This is the 8 to 12 watch. We'll be back on microphone in just a minute, but as we go silent, we'll be conducting a private ceremonial protocol that acknowledges and honors the biocultural resources that are present and that we'll be interacting with and impacting. So we'll be back on microphones shortly. At this time, I invite everyone in the control van to go ahead and mute your microphones. All right, we've got about, front row, correct me if I'm wrong, 30, 40 minutes to boogie down here before we need to start
something like
hanging
that
in the blue water.
Yeah.
So we are on the Sovereign Seamount. We are working our way east up the western flank of this, which I believe is a conical shaped seamount. I actually can't tell from this view. And we're at a pretty deep depth on today's dive. We're currently a little deeper than 3 kilometers below the water surface. And we're exploring down here to see what kind of signs of life are present at these depths and how they connect to the communities on some of the other seamounts that we've been exploring on this expedition. We've also collected several rock samples for analyses on shore to try to figure out the age of this seamount and the conditions under which it formed. But I believe all the rock samples we need for this dive have already been collected, so our focus today for the remaining 40 minutes is to look for animals. And if we see something that is on our high priority list, we might stop and sample.
Roger. So the ship's come to a stop. I'll just call in another move. We were going 075.
Yeah, that sounds great.
Roughly along the ridge, but kind of hard to judge.
Wonderful. Thanks. A question for our data logger / coral expert. What kind of cool stuff has been collected so far on this this dive? Do you know?
Yeah, we've got a pretty full starboard box full of rocks, at least half a dozen collections or so. And then we've got a number of slurp jars full, probably the most of the crew so far.
Okay.
At least 1 bamboo coral, yellow swimming crinoid, cup coral, and anemone. And then in the forward box, we've got some floaty bits, stalked crinoid, a sea cucumber, and a glass sponge. So,
do we, what is the priority for trying to get an eDNA sample on this deep dive?
Yeah, it's pretty experimental.
Okay.
So anything we take would be kind of just a hypothesis testing to see if we could detect anything. But usually you don't find high density communities below maybe a couple thousand meters. So it might be worthwhile to maybe try one, but yeah, none of the things we've seen on this dive so far would classify as a high density or high diversity community in the traditional sense.
Okay. Video, is there a way to bring up the gain a little bit on the Herc cam? Thank you.
There is. Did you want me to?
It looks like you already did.
Well, good morning to our viewers around the world. We have many, many of you both on nautiluslive.org as well as our YouTube channel. If you're on the nautiluslive.org portal, you can throw in a comment or chat where it says we are available for questions. We're getting some questions about what is the most exciting thing we've seen on this dive, but it feels like a lot of you have been with this dive since it began about 8 hours ago, and I would encourage you, I'm going to throw that back out at you. What's the most exciting thing that you have seen on this dive? Let us know in the chat there.
Yeah, so far for me, it's we've only been here for 5 minutes, so I don't really have a good reference point.
Yeah, the way this works is we do have 4-hour shifts of our scientists. The ROVs can stay underwater pretty much indefinitely. They don't need to sleep, eat, or use the bathroom, unlike humans. So we do 4-hour watches, and I've just swapped out with my counterpart, Afrodita Fuentes, and I'm excited to be here with you today. Let us know if you have any comments or questions, please.
Yeah, and this is going to be a busy morning, too, with we've got our ship-to-shore interactions going full swing this morning, so I know I'll have to be stepping out of the control van for a few moments later in the shift. So our science communication fellows like Lindsay are very busy today, swapping in and out with different tasks.
It's fun. It's great to connect with everyone around the world.
Yeah, and I think we're pretty full up for this expedition's ship to shore, but there's always chances to connect for future expeditions. So if you're joining us for the first time or aren't familiar with these opportunities, if you go to the nautiluslive.org website, to the education tab, there's information there about how to request a ship to shore interaction for your classroom or other learning groups. And it's a chance to interact in real time, ask questions of the team in a one-on-one setting. Can we get a partial zoom on that sponge there, please? And Rennie, could I ask you to zoom out on HIPAC to give a broader view of where we are today?
So we've gone past our dive-in target, just continuing up the slope. We're at a less steep portion of the slope.
Yeah. Thank you. I'm not good at sponge identification, so do we have any guesses from the back row of what we're looking at?
Definitely a glass sponge.
Yes. Oh, we got a couple good shots. We can keep moving, Jess.
Sorry, I was off SPL. Roger that. Yeah, so this is a fairly deep dive. Steve, is this too deep for cephalopods, jellies, and fish?
Not necessarily too deep. Things are just more sparse. Typically, the deeper you go, the less dense you find animal life because of just limitations in food supply in the form of marine snow with good, high organic carbon content. So for the most part, I think, typically the deeper we conduct operations, the more time you have to spend on the seafloor to see things, and you really just get a fraction of a snapshot when you're down here for a few hours. So this is really kind of more of a reconnaissance of the area and the depth than a true kind of survey.
Viewer says that their favorite moment was when the ROVs first reached the seafloor, and one of the watch folks said this was the first time humans had set eyes on this particular seabed. They had not realized watching the earlier dives that no one had been here yet to explore, and is a good reminder that we are some of the first eyes to see these rocks.
That's right.
Is that a holothurian there? Or a Walteria?
Oh, yeah. Or in the 3 o'clock position.
Oh, yeah, okay. Kind of falling over there.
Maybe it's nothing.
But yeah, come back to your comment, Lindsay. That's correct, that every site we're visiting on this expedition, we are the first visual documentation and exploration of this part of the world's seafloor. Oh yeah, definitely not a Walteria.
I go wide, please.
Very nice.
There was some kind of pro-move crinoid collection that happened in the last watch that is very popular.
Oh yeah, I did see that as I came up for breakfast.
I think I heard some cheering when I was in my interaction. So.
Yeah, I think I saw some arms wave in the sky behind you.
Was it one of those big red ones that we just saw as we popped on watch?
It was kind of more of a yellowish, a goldish color. Yeah, involved a little snipping and slurping.
Been apparently lots of tunicates spotted on this dive, but not as many of the little anemones that our watch saw yesterday. It is interesting how each seamount has a slightly different, or at least each ridge on the seamount has a slightly different personality.
Yeah, well, and I'm sure that if we, one of the things we saw yesterday on the dive at Euphemia Seamount was, you know, we started on a more southern ridge and then ended up on a more northern ridge and got a chance to actually, unplanned, do a comparison of how one ridge looked different than another ridge. And we've also seen that at Don Quixote Seamount, where we did dives on multiple ridges coming off of that guyot, and saw that there were definitely big differences in the density of the animals that we saw. Yeah. So I'm sure that if we picked another flank of the seamount we're on today, Sovereign Seamount, we would maybe see those differences, although as Steve pointed out, we're kind of just skimming the surface of these really deep environments where the density is a bit more sparse, and so really getting quantitative differences may be a challenge in a spot like this. Because we are pretty deep for this area, a little over 3 kilometers water depth, or about 2 miles down below. For our audience that has a better reference point for miles than kilometers. Lindsay, do we have anybody joining us on Nautilus Live who's tuning in from Hawaii?
We do have a number of folks from Hawaii today. Yeah, aloha.
Yeah, aloha and good morning.
We have actually quite a number from Hawaii today.
Great.
And our watch is really fortunate and lucky to have Kalamehu here with us as our Hawaiian cultural liaison. Aloha, Koyaka.
Aloha. Yeah, no kapoe, no na kanaka, no na leho leho, na mama aloha o keia mau aina, aloha pumihana kako. Ina oko he poe o lelo Hawaii ma ke a he ninawa he mana o paha ko ke olo olo mai, ho'ona mai, ninau mai, keu hoi ka poe kamali I nana puana ma ka lumi papa pela aku. Ina oko nana mai ana ke ia wahi po kalamu, kama ko hana ma papa hana mo kua ke nei, ke olu olu, ho'olaha I ka lehu lehu. O iai, ai no kahi poe, pa ana ko lelo Hawaii malama puana, ke la mau iki ana kupuna ma nei nei, aloha pumihana.
Any chance to zoom on that coral in the upper right, the V-shaped branching one? Just off screen now.
Yeah, it's just up here. These are some really nice basalts here that we're looking at too.
Looks like a primnode from here.
Go ahead and push on in there a bit, please, Jeff. One of the other things we're seeing in this field of view is some what look like little black trails on the rocks. A little wide. And thank you to one of our scientists ashore for sharing some information about what the potential origin is of those. So on previous dives out in the
let's get there, I think.
Previous deep sea dives, these kinds of trails were attributed to a limpet. So it might be the same case here, but a little hard to tell when we don't have the animal present. We just have the indication of where it was.
All right, full wide there, Jeff.
Could it have been the chitons that we've been seeing? The Polypocora?
Potentially. I don't know.
Some kind of mollusk?
Yeah, it's kind of a guess.
Is that a tunicate? What
yeah, it looks like there's a tunicate falling down on that.
Yeah, sorry, guys.
Yeah, you got a boogie?
Yeah.
Okay.
Push out. But we'll probably see some more. I heard they're seeing lots of tunicates. Is that the...
So we need to come up with an alliterative team name for today's shift.
Oh,
maybe it needs to involve tunicates. Totally tunicates.
I have a question about where is the chat.
Okay.
But you have found it. If you typed in this, you found the chat. It's actually...
Oh, Jess?
Yeah?
While we're here, can we get some shots of these ripples in the sand? That's a request from one of our scientists.
Sure thing.
And if possible, from a couple different angles. I know that's going to be difficult as we're on the move.
Yeah, I'll do a...
They're really trying to kind of see.
Can I go ahead and push on in a bit, please, Jeff?
Angles of the ridges here.
Exactly, yeah. So there's an interest in trying to understand the topology and formation of these ripples. And so getting the image in a couple different fields of view from different headings can help understand that a little better. If we observe something like this again in a situation where we don't need to be on the move, we would also try to collect a sample. But I think in this case,
yeah,
since we need to catch up, that's not going to be possible.
Are those good? Are those shots all right for that?
Those are great, Jess. Thank you.
Full wide.
Gonna do a DVL reset.
Roger that.
Beth, the question about some of the samples that you've been processing, and maybe this is for Steve as well, just wondering if there's been any evidence of microplastics or if that's something that you're seeing residue of human impact.
Yeah, that's a wonderful question in terms of different things that scientists should be on the lookout for while they're processing samples, even if that's not their primary question. In the water samples that we have collected, there haven't been visible signs of visible plastics, but that's something that can maybe be looked at on the filters back in the lab for, micro and nanoplastics. And some of the samples that I've been collecting could also be used for interrogating that, but I imagine the densities would be relatively low. For the animals we've collected, I don't know that we've done like a gut inventory. Well, I know we haven't done like gut inventory out here, but that could be something that scientists ashore could maybe look at. So no visible signs, but it is something that could be investigated further.
Cool, thank you. We have a medical professional watching who's wondering if any medical research or info could be gained from these deep water sponges and ground samples.
Yeah, potentially. So there are sponges are known to have really interesting kind of chemical defenses as part of their microbiome. And there are several teams that are exploring the sponge microbiome for potential compounds.
All right, Jeff, go ahead and push on in there,
please. Oh, look at that. Compounds of pharmaceutical use, potential anti-cancer compounds, things like that. I don't know that deep sea corals have been investigated in that same way, but I wouldn't be surprised.
Yeah, they have.
We're getting the affirmative that yes, they have.
Yeah, great. So that could be something that's explored. As part of our permit for this work in the Papahanaumokuakea Marine National Monument, we are not permitted to directly use these samples for bioprospecting, which would be the typical case for that kind of research. If researchers wanted to do that with the samples that are being collected and sent to archives, they would need to request special permission from the monument management. So that isn't going to be a primary focus of the research of this expedition. And folks listening online may be aware that one connection of deep sea microbiome biotech connectivity is in relation to some of the enzymes that have been derived from organisms from hydrothermal systems have been used in as enzymes for some of the research being done on COVID-19 and identifying viral sequences. So there is some connectivity of deep sea research to broader questions. Oh, look at that beautiful. Yeah, maybe that is the kind that was sampled earlier.
Yep.
So it does look more red here when it was being collected. At least the view upstairs, it looked more golden.
You gonna do a partial here, Jeff?
You can see actually this one. You can see the gastropods. There are some small snails that graze.
Very small.
On the pinnules and branches of these crinoids.
All right, full wide, please.
Thanks, Jess.
They look like small white dots. I saw those a few minutes ago as well before the watch change.
The sample that was collected right before the shift changed, was there evidence of those same gastropods on it?
The stalked crinoid was collected a while ago. I actually didn't see it.
Okay.
But we'll take a look at it. I mean, it would be pretty interesting, noteworthy find to get some of those associates.
Just a heads up to Steve and Kalamihu, I'm back in the log.
Okay.
Oh, great.
Although I'm loving seeing Kalamihu take on the science logging role. Look at this. Made you a scientist.
He's actually helping us with the story highlights.
Yeah. It's awesome.
Science.
Here's another one of these stock crinoids coming into view. Almost looks like a little fireworks greeting our morning here. Seems that there's maybe an anemone in the foreground or the background.
That anemone looks like a little crown, doesn't it?
Mm-hmm.
Dr. Beth, you've been talked about in previous watches, your pioneering work in the deep sea field of sea microbiology. Wondering how much of our perspective on the marine ecosystem as a whole is expected to change as we learn more about deep sea microbial life. Also, good luck on the rest of the dive.
Oh yeah, thanks. Yeah, that's a fun question to think about. You know, scientists have been studying the deep sea for several decades, as these kinds of technologies like crewed submersibles and remotely operated vehicles have allowed us access to these environments. You know, we have sometimes a baseline understanding of the microscopic realm of life that lives in these environments. But one of the things that's a focus of my current work now is to try to think about that in a frame of reference about the kind of connectivity and service that the microbial realm provides to the larger ecosystems, because there's increasing human interest in accessing potential resources on the seafloor. And so that could lead to more perturbation of these environments. And so trying to get a better understanding of what is the natural variability of microbial communities, how resilient are they to impacts, what are thresholds of harm? You know, one of the things that guides a lot of work for deep sea research in the biological realm is in thinking about different species. You know, do we see coral species X, coral species Y? Those definitions for microbes are a little bit more squishy, and so does it really matter if you have exactly the same species as long as the function is still being carried out? And that's a really open question in microbial ecology, not just in the deep sea, but in many environments. Do we want to get a partial zoom on that, Steve?
Yeah.
Can we get a partial zoom here, please?
Yeah, sure thing.
One more request too. If we are ahead and we're in a good place, can we sit down next to that coral and take an skin sample?
Sure thing.
Sounds good.
And then we're doing the math back or up here, I guess. And yeah. It seems like we'll only have about 5-10 minutes left of this dive, and then we'll start getting set up. Go ahead.